All right, so let's go with our pre-recorded lecture here on transcription translation. So just a little review on what's happening with DNA, or deoxyribonucleic acid. It's that double helix form that has both adenine and thymine that pair together, complementary, and cytosine and guanine. There are four different nitrogenous bases that match up with each other. Adenine and guanine are in this one group, and thymine and cytosine are these other group, but adenine binds with thymine and cytosine binds with guanine. And I believe that we have the purines, which are Ag, and the pyridamines, which are thymine and cytosine. But you don't have to have that memorized. Just know that they're in different groups, and it's because these ones have the two rings, and these ones have one. As we talk about DNA replication, it goes in a specific direction. It also goes with a DNA polymerase that is going to read the template strand and make a complementary strand based off the existing base pairs that are there, nitrogenous bases. So it matches up, and DNA polymerase will also proofread as it goes through it. It creates these replication forks at different places. So you'll have a lot of different spots on a DNA strand that are getting copied simultaneously. This is just showing how you have to first unwind and unzip with the use of helicase. Then once it's exposed and you have the DNA that's open, like remember, the reason it is actually double-stranded and the reason that it's closed up like this is it protects it. This means that there's a less opportunity for it to get damaged. So of course you have to open it. So now it's a little bit more vulnerable, but at the same point you'll be able to have DNA polymerase to match up the appropriate base pairs together. And then once you have it formed, you have your two new strands. And each strand is made up of half of the same information, and it is semi-conservative. So now that we've just kind of gone from that, we're now making our photocopy. If you guys remember when I said that you have your recipe book for the cookie in the nucleus, you need to make a copy of it so that it can go out to the recipe that you want, so you can go to the cookie factory. This essentially is transcription. RNA is your photocopy. There's some things that make it unique. It's single-stranded. It uses uracil instead of thymine. It has ribose instead of deoxyribose, and it leaves a nucleus. It's also shorter. It's a smaller version, right? It's not the whole thing, so it's a lot shorter. So these are the four things that you have to know, right? Single-stranded, has uracil instead of thymine, has ribose instead of deoxyribose, and leaves a nucleus. Right? This is likely to be a question on something. There are three different. So here's and there's three different types of them. We'll look at them. Here we have deoxyribose, which you find in DNA. Here you have ribose, which you find in RNA. What are the differences? Not much, right? If you look at it, it has a hydroxyl group here on this second carbon, and that's pretty much it. But it makes a big difference. So these are the three different types we have. We're going to really focus more on this mRNA because this is the one that's going to be the photocopy that's coming out of the DNA. We have ribosomal RNA, which is used to make ribosomes. So if you guys remember, ribosomes are the structure that is used to make proteins. They are made up of rRNA plus a protein. And we have transfer RNA, or tRNA, which happens during translation, which alongside with the ribosome will read the mRNA to make a protein or a polypeptide, and eventually the polypeptide will turn into a protein. So looking right here, we have transcription going on. It goes in the 5-3 direction. This just has to deal where the phosphate group is on it, and as it's getting copied, same idea. You have RNA polymerase, except instead of having helicase and then DNA polymerase, RNA polymerase basically does both jobs. It unwinds and unzips and copies at the same time. One thing that's important to note on this, right, is that you have to know when to start doing transcription, and there are special things that tell RNA polymerase, hey, I need to go to this part of the DNA and copy this part of the DNA, and this is where the signals come in from the cell. It's kind of crazy that this happens, and it's interesting that it happens as well. But notwithstanding, this is what happens. RNA polymerase opens the gene, or the part of the DNA it needs copied. It reads it using the same theory of complementary strands. It matches up the nucleotides, right? Remember, RNA and DNA are actually pretty similar in a lot of their basic steps. And anytime there's a T, it matches with an A. but every time there's an A, it matches with a U. This is one of the unique things about RNA. It has uracil instead of thymine. And as a result, anytime A matches up with something, it's not matching up with a T, it's matching up with a U. And that'll be like a clue if I'm asking you, is this a strand of DNA or an RNA? How you would know whether it's DNA or an RNA, right? Shorter, single-stranded, U instead of T, has ribose instead of deoxyribose, and can leave the nucleus. So here are the steps. First, RNA polymerase binds to the promoter region of DNA and separates the helix. As I said, you have to know when to start making the DNA. And there's something called a promoter region. We aren't going to get into the details of it, but just realize it's a promoter. It's telling you, hey, let's get this started. Then RNA polymerase will start matching up the nucleotides. And instead of using T, it's going to use uracil. Then, after it's finished being made, it leaves the nucleus. So this is what they always talk about as what's called the central dogma of molecular biology. DNA becomes genotype, it becomes a phenotype. Or it goes DNA to mRNA to a protein, essentially, right? And this is the magic part where you're basically making DNA into a trait, which we're going to kind of really work on today, or we'll really work on with our rebop. So let's check this out. You guys will have to, do this on your own because I cannot show it individually. I'll put a link inside there because the screencast app doesn't allow me to share my audio sound.
Here is a cell.
So summary of, well, maybe we can watch it. Let's give it a shot.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, so summary of protein synthesis, right? If I was going to be drawing this out, where is my annotator? Why can't I annotate? Nope. Pointer. Sorry, I guess I normally annotate with a different program. So let's go on to this right here. Basically, this is a summary of protein synthesis. In protein synthesis, you copy the part of the DNA that codes for a gene or a protein. This happens as called transcription. RNA polymerase reads the DNA and creates what's going to be eventually the copy that's gets sent out to be made a protein of. There is a process where it's called where you have to it's like processing, DNA RNA processing, where you have to remove introns and keep the exons, and then you have what's called this mature mRNA. We're not going to focus on that too much, but just realize it. Then the mRNA leaves. You have this ribosome, which basically kind of clamps down on it and reads the codes 3 at a time using a codon, and then matches it up with a tRNA that is carrying a amino acid. So you get a chain of amino acids, which is building your protein. So it's a little bit tricky, right? Because we're using a lot of different words here, but we have this ribosome, we have this mRNA. Making the mRNA is just a photocopy of the gene. What we need is this ribosome to go ahead and bring in all the tRNA. Now each tRNA has a specific anticodon, and that anticodon corresponds to a 3-letter sequence on the mRNA, which is called a codon. Each tRNA with each anticodon is specifically holding a specific amino acid. So you can see that each 3-letter sequence codes for a specific amino acid, and proteins are made up of specific amino acids. So it uses this as a way to know which amino acid to bring so that the protein is made up of the correct amino acid sequence. The mRNA attaches to the ribosome. Then it starts bringing in these tRNAs to match up. G with C, C with G, A with U, of course, because the tRNA, which is right here, is made up of RNA. So of course it doesn't have a T, it has a U. Then the tRNA brings the correct amino acid to the ribosome, and then they match up until it reaches the stop codon, and then the ribosome will release the mRNA, and then it'll go for post-translational modification. This is how mRNA codes for specific amino acids. These on the outside are the amino acids. These on the insides are the different combinations of nucleotides that will code for one. So for example, if I'm trying to make valine, I need to have the code GUU, GUC, GUA, or GUG. All 3 of those will code for valine. If I'm hitting a stop codon, right, these black ones are stop, UAA. That means that it's time for me, or the ribosome, to let go of the mRNA. Methionine is a start codon. So each gene starts with an AUG. Well, once it's an mRNA, starts there to let you know, hey, it's time to start coding for an amino acid. So this is how, start coding for a protein. This is how you know that a protein is being made. I have the amino acids DNA versus RNA, and that's it for now.